So just to recapitulate, if you weren't here last evening, in Australia in 1975, there was a lot of confusion. Worldwide, there was a lot of confusion about the brittle bone disorders. And in 1972, a group of people with genetic bone disorders and primarily short stature funded a study to find everybody with genetic short stature in Victoria. And out of that, there were about 300 people, and half of them had brittle bone disorders. So brittle bone disorders are the most prevalent as a group of all the genetic bone disorders in the community, and everything else is rare or ultra rare. So there may be disorders where there's just one person, with that particular disorder. You can hear me presumably? It's all right, Fern, I might just... Is that better? That's better. Yeah, I think that's better anyway. So this brings it up to the study. So I was asked as a young fellow with a colleague, Alison Sen, who was an honours student, we were asked to look at the people with brittle bone disorders. And in fact, we ascertained, including all the babies who'd passed away since 1965, I think it was, or 55. It was about a 20-year retrospective. We ascertained everybody with OI, and that was about 180 people. And we drove all around Victoria to meet them. So we met people like Rosie from Kyabram and, the sports writer for the Telegraph who lived in Bendigo and things like that. Now that was actually really important because then I went away and studied. We wrote some papers. We weren't expecting a huge amount to come out of this, but I pursued my studies in genetics with the leading group in the United States. But where we are today is with a group of brittle bone disorders, collectively called osteo, meaning bone, genesis, bone forming, imperfecta. That's at least 3 patterns of inheritance, and I'm going to talk a little about them because it's time that we recapitulated, because they're important to how you go about looking after people and also treating them. And we broke these down into 5 groups of disorders, and they were previously called OI types 1 to 5, and I guess we'll stick with that because it's easier. And there's now 26 genes implicated in causing different types of OI, and I'm sure there'll be some more because I have a number of friends who continue to work on discovering the mechanisms in certain families that are not yet published, and there are 9 rare syndromic types. So we gave these names to the disorders, but they became, in the literature, they're written as Roman numerals. Now, the International Nomenclature Committee recommended that we don't use the Roman numerals because with the naming in the genetic database up to 19, it's very complex, and unless you're good at 14, 15, 16, 17, 18 and 19 in Roman numerals, which most people aren't, you will get confused, particularly when we get on to 21, 22, 23. So anyway, so they recommended that we just use Arabic numerals, and that's OI type 1, and there's 2 subtypes. The type we call common variable type with normal sclerae, people who don't have the distinctly blue sclerae. There's 10 different subtypes, and as you can see with what they call the deforming types, the tendency to deforming types, because some people have no deformity despite the fact that they have inherited a mutation which might lead to a deforming type. There are 19 different subtypes. And there were 6 types that led to babies dying at birth or within the first year of life, and they were called OI type 2. And if we get some time, we'll talk about this. So the OI Australia, or well actually OI New South Wales back in 1981 funded Dr. Christine Barlow Stewart, who went on to develop the Centre for Medical Genetics in New South Wales, genetic counselling in New South Wales, the profession of genetic counselling in Australia. Chris helped me ascertain the people in New South Wales with OI. And if you look at the bottom 3 lines, we ascertained that there were 530 people over this 30-year period with OI in New South Wales, and 378 over 16 years. And there were, what was important here was there was 101 young people in transition, many for whom there was really uncoordinated services. So a lot of people in the community with OI, and many of them have been and come and gone as members of this society. So what are these disorders? These disorders are genetic disorders, heritable disorders, and there are a number of genetic mechanisms. There are some rare genetic mechanisms, which I'm not going to go into with your group, but to tell you there are people with complex deletions within their genome where they have deleted the whole of a gene. Remind you that we're made up of billions of cells, and every cell has this structure with, and I forgot to bring my pointer, but just to point out that cells have a nucleus in the centre, and that nucleus has a membrane around it, and it contains this substance called DNA. And when cells are dividing, we can look under the microscope and we can actually see the DNA all packaged up. It's all coiled up like a skein of wool. And then with various special stains, and this is black and white photography, but with special stains it looks like this. And we find that in humans we have 46 packages, and there are 23 pairs of these packages. And men and women, as you see here at the bottom, have a difference, and women have 2 X sex chromosomes and men just have this X and what's called a Y chromosome. So those chromosomes are actually like a coiled, they're coiled up thread of deoxyribonucleic acid. So the chromosome, and then if you go down further, the coiled thread is 2 strands or double-stranded, and those strands are linked together through very special bonds, which are between the 2 substances, adenine and thymine, and guanine and cytosine. And this is the story, you know, of DNA that Rosalind Franklin predicted, and Watson Crick stole and published and got the Nobel Prize for. Anyway, so guanine and cytosine and adenine and thymine, and these 4 bases, these 4 letters, you know, make up the whole. So there are packages of them like triplets that's code for amino acids. There are sequences them which are punctuation. They punctuate the whole message, the whole library. And here you can see another cartoon of the DNA all bound together, and A binds with T and G binds with C, and it must always be that particular rule. So in most people with OI, the brittle bone disorder results from spelling mistakes in this DNA sequence. So there are various types of spelling mistakes. There are spelling mistakes which lead to a missense. In other words, here you can see in the top that follow the arrow is that there is an A replaced by a C, and that changes one of the building blocks of protein, glutamine, into an asparagine, and that in itself could be the cause of a genetic disorder. And then there are what we call silent mutations, so they are substitutions of nucleotides which don't change the amino acid at all, from here a glutamine to a glutamine. And then these very important mutations that if this spelling changes, and here you can see a spelling of a G to a T at the bottom, and there are certain signals that are called stop signals, and those signals are generated by TAA and TAG. So any substitutions which change the sequence to those leads to a stop. And then there are frame shifts where we have insertions or deletions. So there are whole different types. Of course, there are in-frame insertions and deletions. In-frame are deletions or insertions that are triplets of 3, and frame shifts are shift 1 or 2 or any other number which is indivisible by 3 leads to a frame shift. So what are we doing today? I'm not going to go and give you a lecture on all the clever machines. New South Wales spent $12 billion to buy one of these sequences for the Garvan Institute to make a service where we could do 50 genomes a week. And it's all still being sorted out, and there's commercial involvement, and many overseas groups have set up to do sequencing. And these machines just work 24/7, in other words, every minute of the day, 365 days of the year, except when somebody has to change the chemical components to keep them running. But they're in arrays, they're in banks. So, but before we start sequencing, and today we may sequence all the 26, like if we have somebody with a brittle bone disorder, we might sequence the whole genome and then just look at those 26 genes in detail to see if we can see the sequence variant. But before we start sequencing, it's handy to have an idea of the possible type of inheritance we're looking for, and the particular disorder we're looking to solve. So there are these classic patterns of inheritance which were identified over 150 years ago, and these are dominant inheritance through autosomes, and both sexes are affected. Men transmit to their sons as well as to their daughters, and 50% on average of children are affected. And here you see a pattern where an affected father transmits 50% of his sperms, could effectively fertilise a baby, and 50% of the offspring would be affected. And similarly with the mothers, 50% of the eggs. And here's a 3-generation family. In fact, everybody is affected in various ways. Grandfather with his marked hearing impairment, mother and their daughter. And it's really quite, I'm sorry I don't have a pointer, and I don't think the pointer you can see. It does work. Can you see the pointer? Oh, I'm sorry, okay. So you can see here that the change here says in the type 1 collagen alpha-1 chain, in the sequence at position 1703, there's a deletion of a C. That's simple. So that's a frame shift because it's not 3, 6 or 9, and it's a deletion. So what does it do? What it does is it changes the proline, sorry, just lost it, proline 568, and there's a frame shift, and 12 positions downstream, it leads to a stop. So, this family has a heterozygous, only 1 copy, so going backwards, heterozygous, and it's a pathogenic variant. They also have a unclassified variant. So that results in their OI type 1. And these people all have various changes within their sequence. They're not related. There are 3 people with OI type 1 of this nature, which is either a stop, a frame shift, or a splicing error. I wanted to talk just really quickly about OI type 5. It's sort of rare, but probably not that rare, but just missed in the general big group of people with OI. And this disorder was described in 1908, and in fact as I was coming into learning about OI, Dr. Balls, who's an orthopaedic surgeon, was then from Adelaide. He had studied all the people in Oxford and found a number of families who had OI type 5. And they have a very special change, which leads to a variant which is recognised as a start signal. But as you can see, it's 3 times 5, 15 nucleotides, 15 A's, C's and T's upstream. So it inserts 5 amino acids into the encoded protein. So they have an extra long protein. So this is a novel start site, 15 base pairs upstream. So this is a very interesting and rare disorder, and it's picked very quickly by looking, for example, this young lass here. You can see if you look at her elbows, my colleague who looked after her as a baby called me up and said, have you ever seen anybody with OI with dislocated elbows at birth? And that's very characteristic. So these 2 young people, you know, have this OI type 5. And it has very special characteristics. So they develop bone that fuses their 2 forearm bones between them, and you can see the bone forming. There it is there, the extra bone forming between the radius and ulna, or the ulna and the radius. Okay, all right. So autosomal recessive, there's 19 different genetic causes of OI type 3, and many of the recessive disorders result in a progressive deforming phenotype. And here both parents are carriers, and many carriers in the normal population, but if carriers for the same rare variant come to have children, they have, by the law of the odds, a 1 in 4 chance, or 25% chance, that the child will receive the variant from both parents. So one of the disorders that we became very interested in our clinic is the group of disorders with OI, with brittle bones, with joint contractures, and they often have these tight arms and knees, you know, at birth. And there are 2 types of this so-called Brooke syndrome. One has mutations in a gene called PLOD2 and the other in FKB10. And FKBP10 variant, osteogenesis imperfecta, is the commonest form of osteogenesis imperfecta in southern Africa. So when you go to southern Africa, there are more people with FKBP10, and that's because of founder mutations in their populations, founder recessive mutations, so more people in their populations are carriers. And it so turns out, and this is from Samoa, it so turns out that this is a common disorder in Samoa. In fact, our elder sister in this family that Tim Cundy described, she has the femoral fractures and something called protrusio acetabuli. In other words, her hip is bent inwards, or her pelvis is bent inwards at her right hip, and she's also fractured through her femoral necks. But brother only has the joint contractures, and we have such families in Australia, and it is important to sort out their changes. And my colleagues will tell you about some of the young people we've looked after here. And here's one of our young people, and you can see it's a major orthopaedic challenge to be able to straighten that hip. In fact, remaking a hip with protrusio is virtually impossible. So some 30 years ago, we were told about sex-linked osteogenesis imperfecta. So this mainly affects boys in families. Sisters and mothers are mildly affected. This has been not a subject that's been discussed widely. But there are now 2 types of X-linked OI. And here you can see the pattern is that mothers carry two sex chromosomes, but the genetic variant's only on one chromosome, only on one chromosome, on average 1 in 4 of her sons is affected, and 1 in 4 of her daughters is a carrier, and those carrier girls could go on in families and have them. And here is from Fleur van Dijk's study of PLS3 mutations, and here you can see that there are 2 carrier women. This one has fractures, but very mild, with more severely affected son, and through her daughter she has 3 affected grandsons. So, and recently the other one is due to mutations in MBT PS2. So I'm going to skip over this little bit of genetic counselling, but to say that genetic counsellors are very involved in the whole process of bringing helping families understand what the impact of the genetic variant in their family will be, and they provide supportive counselling. One of the things they are very involved with is reproductive counselling, and this is really very complex. So this is not like cystic fibrosis where there is, perhaps making it sound easy, for CF, a common recessive disorder in the population, where the patterns are fairly similar, for people who are affected, and most people in the community have only this rare type of delta F508 mutation. But in OI, you've got all these possibilities: 26 genes, missense mutations, nonsense mutations, frameshift mutations, and mosaicism. And mosaicism is where a parent actually has a mutation, but it's only in some cells, including reproductive cells. Our other problem is that it's not possible to sequence and interpret all the modifying genes. And it's clear, if you know families well, particularly when you know several generation families, there is a lot of variation within any one family. And we have some good information that quite a bit of that variation is due to the genetic background variation within the family. So what are the options? They are, if a spelling mistake, a variant is found that causes OI, then parents want to know about having children, and some people elect to have no testing. I have the disorder, I don't want any interference. Chorionic villus sampling between 11 and 13 weeks, and then pre-implantation genetic diagnosis. So I'll just jump over that. And here's where I was going to introduce Kate and Jodie Owen. Jodie's not arrived yet, or? Still driving, thank you, Sharina. Well, maybe you could speak for just a couple of seconds, Kate, and tell people. Okay, so.
Some of you already this weekend, and I met a few of you last conference as well at Melbourne, if you guys were there. So I have dominant OI, so autosomal dominant. My father has OI and my brother has OI. We've all got type 1. I was diagnosed at 17 months of age with a tibia fracture. I fell off the lounge, and my mother took me to emergency, where a registrar luckily had a really large brain. And we all know what registrars are like. Sometimes they're very arrogant, sometimes they don't know what they're talking about, sometimes they don't listen or look properly. So that was excellent that they were actually really helpful. And with my father's history, they weren't exactly sure about his OI at that point, because it was early 80s, and he lived on a farm as a child, so obviously, rough and tumble, lots of fractures, used to get smacked a lot by his mother, would sometimes break, but she was a pretty hard woman, so, that was pretty normal for back then. So I met my partner Andrew about, I don't know, close to 10 years ago, and we were pretty adamant that, well, I was always adamant regardless of whether I was having a partner or not, that I wanted a child that, you know, ideally wouldn't have OI. Given my history, I've had multiple fractures of my life. I don't necessarily look like I have, but both scapulars, pelvis into acetabulum, sacrum, yeah, lots of stuff. And if I had a child, I'd want them to play whatever sport they wanted. That was the biggest thing for me, because I had to give up sport by the time I was 14, which was a bit annoying. I was very lucky to play sport up until that age, to be honest. So we went through the process of the pre-implantate genetic diagnosis. So in other words, a bit of an embryo sort. So I can get into the details if people want to later one-on-one with me. I chatted to Fern before the conference, and given it is a very personal choice and it is very sensitive to talk about for a lot of people, and it's understandably, you know, pretty invasive. So I'm very happy to talk one-on-one confidentially with anyone, and my details, I think we'll pop them up on the site for anyone who wants to email me or just get on Messenger or whatever. So Andy and I, it was a 3-year process. We had to go through the standard conversations with genetic counsellors, scientists as well. We went through Genea, which was previously Sydney IVF. Long story short, we ended up with, so I had the egg collection procedure, which wasn't fun, but you know, that happens. Lots of ladies understand that things get called uncomfortable, but you know, let's just say it was more than uncomfortable. But you know, having a strong pain threshold was helpful, and fast-acting fentanyl was also very nice. So we got 8 eggs, 7 of those eggs fertilised. With PGD, they don't like to take any chances. So a lot of pictures of IVF you'll see, you know, where they have the petri dish. They always they chop off the tail of the sperm and manually inject the sperm into each egg, because they don't want, you know, some sperm are lazy apparently, so they're just like hanging around and like, oh, I don't think I'll try to do anything. So anyway, so that's the manual situation there, so it's pretty cool. So 7 of them fertilised. With PGD, it's a numbers game. So understandably, with different types of OI, there's a 50% chance for some people, 25% chance, all different chances. With me, it was definitely a 50% chance of passing it on, but it sort of ended up being less, which was unfortunate for us. So 8 eggs went to 7 embryos. 7 embryos, they have to be of a certain grade to even test for the OI. So out of those 7, only 3 were suitable to test. They need to be of a certain grade in terms of being able to take away a couple of cells to then test the embryos. Out of those 3 embryos that were tested, 2 were actually affected with the OI. So we ended up with 1 embryo in the end, and that's Cooper, and Cooper's with Andrew out in the playroom, which is pretty amazing. So we were extremely lucky, and I think that one of the biggest things that I was probably in a bit of denial about, Andrew is very much a matter-of-fact man and numbers man, and that's why we do so well, because I'm like, oh, everything's going to be so great, and we're going to get, like, out of those 8 eggs, we'll get 4 embryos that are totally fine. No, because the statistics are, unfortunately, the decrease. So you want, obviously, as many eggs as possible, and that's where it's up to the IVF specialist to really be onto your cycle and, you know, knowing you and how you reproduce naturally or what have you. So we were very lucky, but it was a huge process, and you know, financially, physically, emotionally, it's a lot, but 100% worth it for us. So any questions at all? Like I even said to Bernadette, you know, for when her daughter gets a bit older, you know, she can have a chat to me, and I'm always going to be around with the society, so as people get older and if they want to have a chat to me, you know, about the options, that's totally available. I appreciate it's not for everyone, but at the end of the day, if you want to do it, I'm around to have a chat to you. So please feel free to come and chat to me over the weekend or get in contact online. So thanks for listening.
And similarly, Jodie's coming and Jodie's available to speak about her experience also. So last evening, if you weren't here, Professor Craig Munns, who's the endocrinologist and Professor of Bone and Mineral Disorders at the Children's Hospital at Westmead, spoke about bisphosphonates. And in 2019, we hope that most children with OI will today have bisphosphonate modified OI, and all children can be treated who have a low bone density or certain recurrent fractures, except children with the perinatally lethal forms of OI. These are two pictures from Melanie Alcausen's study. She was with us for 3 years. She's in the Philippines and has helped set up the OI Society of the Philippines. And these two young people were treated from birth. And that's the only reason I wanted to show them, because Craig didn't show pictures of young people treated from birth, but these been treated from birth and done well. And this is the difference between 1975, no treatment in Robert on the left, and somebody at the same age with fractures at 15 months of age treated within the first 3 years of life, but treated, and the complete difference that you actually see. Now, I'm not going to give you a long lecture on bisphosphonate-related osteonecrosis of the jaw, an adult disorder, but Sally's coming after Sally Hibbert's coming, I think, after morning tea, and also atypical low bone turnover fractures. And I'm hoping that Jenny Dale-Howell will actually spoke a little about that. There is a study we did, which is a unique study in the world, full stop. And this is the study of compression of the base of the skull called basal impression. I'm not going to give you a lecture, it would take another 20 minutes, but I am going to say that as a result of a study of over 100 people and how it happened and who it happens in, we introduced into our service something called reclined posturing and seating. And we also gave people we thought were at risk early evaluation for treatment with bisphosphonates. The preliminary evidence is that cyclic bisphosphonate prevents progression of basal impression. So we thought that was pretty important. Here's one of the mums, just nursing her baby reclined. So it's not a problem of babies with OI type 1, but it is a type that does affect babies with other types of OI. We introduced Fraser chairs, these reclined chairs. Our physios and OT use them for a whole lot of indications. And then for older children, we've used tumble form chairs. And yes, indeed, this is Hayley sitting in a tumble form chair. Just wanted to say a little thing about severity, because people got very confused because a number of young people in the United States got confused about these numbers. Well, I didn't give OI numbers. Dr. Victor McCusick gave it numbers because he wanted to gene map them, but probably this wonderful man, Victor McCusick, guessed that was heterogeneous, but my other teacher, David Ramoyne, kept saying, look, it's lots and lots of diseases, look at all your pedigrees, because we'd looked at all the Victorian population. So severity is not the same as an OI type, although it often looks like that, but it's determined by many factors. There's some correlation with a particular gene, so some genes, the mutations we know about at the moment lead to severe disease, but now we're doing whole genomes, and there are people who are going and asking for a whole, all their 3 billion base pairs to be sequenced, or 6 billion, or 3 billion they're finding out that they have these variants that predict these diseases, but yet they don't actually have any type of severe or manifest disorder. And so there's a lot of variation that we don't know about, and of course severity is influenced by but we developed definitions for what was mild and definitions for what was moderate. So, and then definitions for what was progressively deforming, and a young boy who used to be a member of this society who's passed away. And if you follow this, it gives you a good indication, and also of people with extremely severe OI, and often a perinatally lethal course. Now, does all this knowledge about all the different types of OI matters? It matters incredibly, and in scientific circles I can give lectures on what actually a particular type of mutation predicts in terms of the severity, but it's not possible to, and you're such a different group with different disorders in this group here, I can't pop all that into your learning at the moment. But Forteo, which is not used in children, or teriparatide in adults, has been studied in a randomised trial by Eric Orwell. And what Eric and his colleagues, including Brendan Lee, found was that there's a significantly increased bone density and an increase in bone strength in adults with OI type 1 if you give them teriparatide. And it's true that there have been some adults I know well in their 20s, 30s, 40s, 50s, but particularly 40s, 50s and 60s who've been rescued with a year's treatment with Forteo. But it is patients with other types of OI, particularly OI type 3 and 4, it's of no significant benefit. So they studied it quite extensively in vitro and in vivo. Now I think I haven't got time, so I'm going to have to skip over some of this and just talk about research for a moment. So there is what's called a brittle bone consortium in the United States, and they are these centres here. You can see the centres up on the west coast in Portland, Oregon, and Los Angeles, and etc., the Midwest and the East Coast, who are studying various new therapies. And Ingo Graf and Brendan Lee have found and discovered that some mutations in some people with OI have disrupted transforming growth factor beta signalling. And so what you see here are 3 mice. Wild type means mice who don't have OI, mice who have cartilage associated protein knockout, and mice that actually have another type of OI, sorry, the legend's got lost off the top of here. That's a type of OI type 4 mutation. And they've given, if you see the difference between these two here, they've given them a chemical compound called ID11, which modulates transforming growth factor beta. And you can see these streaky bits within the bone of the vertebrae. These are what are called trabeculae, and ID11 or blocking transforming beta signalling has completely restored the bone density of the mice. So they're embarking on a study of transforming growth factor beta inhibition with a medicine called fresolimumab. Now, there's one great thing in OI research, and that is that if you can use a medicine off the shelf that's already been used for something else like Panadol, and it's been through clinical trials, which actually Panadol hasn't been, and you can get approval immediately. So this is a process where you can reuse a medicine for some other purpose, or it's called repurposing, then you can get on with your study without doing extensive safety studies. Now, just very quickly, what's happening over this program here, and this was partly stimulated, Ian and Julie came to the international meeting, is we realise there are some really important questions we have to start to address for adults. One of these are in young adults and adults is that spinal complications may progress, and there are new types of spinal complications such as spondylolisthesis. Respiratory problems may be developed, and Dr. Bristy Roy is going to speak to you this morning from the Westmead group. And obesity is an ever-present challenge, and we're learning more about these things. Now, just a quick thing about the Osteogenesis Imperfecta Societies, our friends from the past, from Jenny's farewell to OI Australia, with OI Australia. But in 2020, the international conference is going to be held in Sheffield, hosted by Professor Nick Bishop. And on the day before, on the 4th of September, there is a patient pre-conference which will be held in Sheffield. The conference is going to be held at the university student accommodation called The Edge. And it sounds like it will be a really important meeting in which lots of new things are discussed. This is a picture from the OIFA, OI Federation of Europe, for which we're affiliated member, conference in Dubrovnik. In there. And just a final word, because I am very concerned for the people of Asia, is there's a lot of people, they have reduced access to basic orthopaedic surgery services, limited access to rehab, genetic diagnosis apart from the Philippines is very limited, and often they can't do bone densitometry. And pamidronate costs are relatively high. Now, Craig didn't mention that there is another bisphosphonate, neridronate, also known as Nerixia, and this is marketed in China, Taiwan, Hong Kong, and well, not Hong Kong yet, and Macau, and it is under license from a Biogen in Italy, and it is actually the only bisphosphonate which has been subject to the randomised clinical trial that is really demanded by authorities that approve these medicines. So there is a medicine, Nerixia or neridronate. It's more potent than pamidronate, but far less potent and less expensive, and it is the standard European treatment that we hope might be the answer for treating a lot of these people in Asia. Okay, thank you.